My name is Stephan Kadauke. I'm the Associate Director of the Cell-Based Therapy Lab at the Children's Hospital of Philadelphia. And the CBTL provides all cell therapy products to children at CHOP. So any kid that comes in with leukemia and needs a bone marrow transplant, we do that, and we have treated a lot of patients with CliniMACS depletion, I think more than 500 patients. So we're a big Miltenyi shop. We've done it all, 319, TCR alpha beta, CD45RA, and I think we've had our Prodigy for longer than many of you guys. I think our serial number is 155. So I wanted to also say congratulations to Miltenyi for 10 years of Prodigy, and also getting your platform into 6 out of 7 commercial products. That is a big deal, so congratulations, and give Prodigy a hand for that. Okay, this is great. And I think this is consistent with my experience of collaborating with Miltenyi in clinical process development all the way up to IND submission, which has always been fantastic. So my talk is titled Point-of-Care Cell Therapy at CHOP and Beyond. So I'm going to talk a little bit less about science and technology, but more about access, highlighting the sort of work we're doing to bring cost-effective point-of-care cell and gene therapy to our own patients at CHOP, but also into the non-first world and to non-first world countries such as Brazil. So here are my disclosures. Miltenyi supported some of this process development, and they're paying me to talk to you guys here today. I got some research support from Lonza itself, and I also want to say in everything I'm saying, I'm speaking for myself, not CHOP. So we haven't had any lawyers check my slides. So here's what I'm going to talk about today. I wanted to first talk about leukemia in children, then about CD19-directed CAR T cell therapy, briefly talk about manufacturing of CAR T cells, some cost considerations, and then I'm going to talk about the Brazil story. And then at the end, if there's a little bit of time left, I want to talk a little bit about gene therapy for sickle cell disease. So pediatric leukemia, B-cell leukemia is the most common cancer in children. Survival for BLL is actually quite good, about 90% survival. And about 80% of patients, they get chemotherapy and it's cured. So that's great. So they won't have the leukemia come back. However, if you're in the remaining 20% of patients who have relapse disease or are primary refractory, I mean, the first round of chemo didn't actually take care of the leukemia. Prognosis is much worse. And traditionally, only 1 in 3 of these kids were surviving long term. And the traditional way to approach pediatric relapsed or refractory BLL was chemotherapy followed by hematopoietic stem cell transplant. And even though patients would receive this treatment, they wouldn't always survive it because the leukemia might come back, or the patient wouldn't survive the stem cell transplant, which is a procedure that's really, really toxic and has lots of morbidity and mortality. But all this really changed for this very specific patient population, for kids with relapsed or refractory B-cell leukemia, through the advent of CAR T cell therapy 12 years ago. And this has really shifted the landscape for kids with relapse and refractory leukemia. So I can't go anywhere and talk about CAR T cell therapy at CHOP without talking about Emily Whitehead. So Emily was 6 years old when she came to CHOP in 2012, and she had relapsed ALL that wasn't responding to chemotherapy. And her disease was so bad that she wasn't eligible for bone marrow transplant. She was too sick to get a bone marrow transplant, so she was really out of options. But exactly at this time, Steve Grubb's group was about to open an experimental protocol. And she was the first pediatric patient who received CAR T cell therapy. And so she received these CAR T cells and actually developed a really severe inflammatory reaction to the CAR T cells, which we now know as cytokine release syndrome. She got very sick, had a very high fever, had to go to the intensive care unit. And when the fever subsided and all this went away, she woke up and the leukemia was gone and it hasn't come back. So Emily is now more than 12 years out from CAR T treatment and is now considered the first patient cured by CAR T cell therapy. And what's really cool is that she got her treatment at Penn, and now she's a sophomore at the University of Pennsylvania. So in total, CHOP has treated almost 600 patients with CAR T cells by now, and most of them We actually don't make them. Most of them are made by Penn next door in their GMP facility. But we've recently started making our own cells in the lab. And so I'm gonna talk about a little bit about how we make how we started making CAR T cells using the Prodigy platform. So I know I'm preaching to the choir here, but here's why we really like the idea of GMP in a box systems. And so the traditional CAR T manufacturing process starts with collecting T cells from the patient via apheresis, and then you have to purify and activate those cells, then transduce them with the CAR gene, and after transduction, there's an expansion period where the cells grow, and then finally you harvest them and formulate them in buffer and cryopreserve them usually. And this traditional manufacturing requires many machines and many bags, and you need to have highly trained technologists performing many manual steps during cultivation. And a GMP in a box system, such as the Prodigy, they can perform almost all of these steps in one machine, automating the process. And so I wanted to tell you guys a little bit about what it took for us to implement a Prodigy CAR T process in our lab, noting that we didn't actually have a traditional manufacturing process beforehand. So one thing that's sort of remarkable is that we had our first manufacturing run in 2019 and then didn't have another one for almost 2 years. And that was because a lot of time had to go into developing sort of FDA-required release tests that we just didn't have at the time. So the ones that took the longest were mycoplasma and flow cytometry. Actually, flow cytometry for CD19 CARs on CAR T cells is what took the longest time. And at the same time, contracting with outside labs and and getting our lawyers to agree on contracts with outside firms that really took a long time, writing SOPs. And this is the part that as I'm going to talk about a little bit in Brazil, where we're transferring this technology to, that we can hopefully leapfrog. So maybe it's not going to take 2 years between engineering run number 1 and number 2. The other thing I like to mention is that we did, which is sort of embarrassing, coming from GMP, is that we did face issues with bacterial contamination, which we could very clearly trace to the use of a water bath to thaw cells. Now, lots of cell therapy labs have water baths, and so we've completely eliminated this and haven't had any more contamination issues since. So yeah, that's something that I like to bring up because I see a lot of water baths. And so at this point, we've successfully manufactured 28 products at a cadence of about 1 product every 3 weeks. So I didn't talk about how big our lab is, but we have 10 staff, and like I said, we provide all cell therapies to the whole hospital. So this is actually, I think, it's pretty good. For outcomes, we say, I think we've infused 21 patients at this time. We see 28-day efficacy, although I can't really talk too much about the outcomes. We see 28-day efficacy similar to commercial CD19 CAR T products. So far, we're very optimistic about the long-term efficacy of these CAR T cells, but it'll take another year or so to really say anything about how they compare to traditional CAR T cells. And there's also an abstract at ASH that I don't want to, from the study PI, which is not me, and so I don't want to spill the beans for that. So just to recap some of these ideas, or the kinds of things that I like to talk about when I come and advise on, how do we set up a Prodigy CAR T manufacturing operation. Don't use a water bath. A method development can take a long time, and only if you need a quick turnaround, do the test in house. Otherwise, see if you can farm it out. One thing that was also really important that a lot of scientists that do this preclinical development is that they do time points that are going to be incredibly inconvenient for your staff to replicate in the clinical lab. So your cells don't care if the hit 1 and the hit 2 are exactly 12 hours apart, but that makes for incredibly terrible hours. So I always say, develop a process that can be performed during reasonable working hours, and also have reasonable tolerances in the timing of your steps. Another question that comes up a lot is which tests need to be. Which test does the FDA expect and what level of validation? So there's actually pretty clear guidance on this, that for early phase studies, the safety tests are the ones that they really care about. They should be well validated. It's not necessarily required for other tests, such as potency tests. So we, took all these lessons learned and put them into the Jovi manuscript and video. So this goes beyond the TCT manual that comes with your Prodigy, but also goes into kind of what our interactions with the agency have been, and how you might modify, when you're setting up your own clinical process development for a CAR with the Prodigy. So anybody who is interested in this, I invite you to check this out. And this is definitely one of those papers that are discussing flexible 7, 8, 10, 12 manufacturing processes for clinical applications, which I think are now pretty common. Like I alluded to earlier, we have taken this process on the road. And this is actually incredibly exciting to me. I actually just came back from Brazil 2 days ago, because we kicked off what they call the Carioca initiative. And for those of you who don't know about this, Carioca is a name that for a person from Rio de Janeiro. And so the idea here was to bring cost-effective CAR T for pediatric leukemia to Rio de Janeiro at the Instituto Nacional de Câncer, the INCA, the National Cancer Institute. And it's funded by the Brazilian Ministry of Health for about $4 million US, which is 20 million reals. And the idea is, or the aim is to achieve a technology transfer of our Prodigy CAR T manufacturing protocol based on our protocols and our SOPs, including the development of release testing and submission to Anvisa, which is the Brazilian health authority comparable to the FDA. We got enough funding to start to perform a phase 1/2 trial of Prodigy CAR Ts for relapsed refractory pediatric BLL patients. And here's an image of here's just some pictures. This is the Carioca initiative. Here you can see some of the key collaborators. Is Steve Grupp on the side, and this is Martin Bonamino, the principal investigator, the head of the CAR T program there, along with a few people from his lab. This is Yongpeng Wang, my lab co-director. Down here, we have actually the director of the National Cancer Institute is talking to us about resources that we'll need to pull this off. Here, these two ladies here are VPs of global strategy at CHOP, and here you see Steve Grupp again. And here we are in the lab, and you can see the Prodigy because we were able to do a 3-day event where we didn't only talk to all the necessary stakeholders when you're trying to stand up a CAR T program, a clinical CAR T program, including the ICU, BMT, pharmacy. And the lab, obviously, but we were also able to perform the first engineering run there. And we got a dry bath into their lab, so they didn't have to use their water bath, which was a huge success. So I want to shift. This is related to this Carioca initiative because really, scientifically, what we're doing is not novel. We're making a CD19 CAR. Okay, these things have been around for a while, but the reason why we were successful in getting funding from the Brazilian Ministry of Health for this is because we said we were going to be able to do it for much am than the commercial products. And here I just want to say CHOP has a very close relationship with Novartis. So when I'm saying that your guys' CAR T product is very expensive, please don't take it personally. Also, this is me talking, not CHOP. Okay, so I don't want to get into trouble here. But the fact of the matter, though, is that if you're looking at the price of FDA-approved cell and gene therapies, such as Yescarta and Kymriah, at almost $500,000 a piece, then the hemoglobin gene therapies that have and other gene therapies that have come out are even much more expensive. Then really what you can do. What we've noticed we can do, what we found we can do, is we can do this for much cheaper. And I just want to make some qualifications here. This is this $30,000 figure. This is the marginal cost of making one patient product, one patient's worth of manufacture, inside of an existing facility, such as a cell therapy lab in a hospital, and you have access to a hospital lab that can do some of the required testing, and it's the marginal cost. So we're not including the cost of the machine, we're not including the cost of the facility. We are including the cost of all the release testing, we are including the cost of the vector, which is expensive, and we are including the cost of labor. So this is marginal. It's probably a little bit higher now because vectors became more expensive, but it's still a far cry from the cost of the commercial products. And I think this is really, when we're talking about patient access, this is really the problem that we need to solve. And this is what sort of gets me up and excited in the morning, is to see how we can get there. Brief recap. We can treat pediatric leukemia successfully with CD19-directed CAR T cells, even for patients with refractory disease. And using a GMP in a box approach with the Prodigy has allowed us to do this at a cost significantly lower than the commercial products. But there's still major challenges, include process development. I didn't talk about this, but obtaining lentiviral vector is tricky, or at least used to be tricky. It's actually easier now. Interactions with regulatory agencies. And that we're actively implementing the CHOP process at Inca, and actually hope to start treating patients in the next 6 months or so. So if I have a few more minutes, I'd really love to talk a little bit about sickle cell disease. So sickle cell disease is a genetic disorder that causes abnormal hemoglobin called hemoglobin S, and it leads to sickle-shaped red blood cells, and these result in vasoocclusion and hemolysis. This affects about 100,000 people in the USA, and they're predominantly African Americans. It's also highly prevalent in Brazil, particularly in the Bahia, in a northeast state, where there are 3,500 new cases per year, according to a recent study. Median survival. So sickle cell disease is not a benign disease. Median survival in the US is approximately 20 years shorter than the general population. In Brazil, a recent study suggested it's actually 37 years shorter. So you're losing 37 years of your lifespan if you have this. Current treatments include small molecule drugs like hydroxyurea, blood transfusions, and curative options for those who have access to them now include hematopoietic stem cell transplantation and gene therapy. So 2 autologous HSC gene therapies were FDA approved in December of 2023. This is Casgevy, exa-cel by Vertex, and Lyfgenia by Bluebird Bio. Maybe one of you guys can tell me how I pronounce that correctly, because I don't know how to pronounce it. Anyways, so Casgevy is based on CRISPR-Cas9 targeting the promoter of a gene called BCL11A. What that does is that it leads to a rearrangement of the beta globin locus, which contains the mutated sickle gene, turning the fetal version of hemoglobin on, and at the same time turning down the sickle globin gene at the same time. And the cost is $2.2 million per patient, and this is just for the product, not for all the hospitalization and all of the other support that you'll need to get the product. So this is just for the product. Lyfgenia uses a lentiviral approach similar to how we make CAR T cells. In addition of a version of the regular hemoglobin that has a single point variant that makes it a little bit more efficient, T87Q, cost is $3.1 million US. So one of the things I wanted to point out that both of these methods introduce double-stranded breaks in the genome to CRISPR-Cas9 targeting this regulatory element works by really beating up on this regulatory element, cutting it multiple times until it's no longer recognizable to the small guide RNA. So there's lots of cutting. And lentiviral addition involves this thing, maybe a less dramatic double-stranded break. But in order to get the lentiviral genome into your host genome, you got to cut the DNA, and this is something that worries the FDA. And honestly, I think maybe that's justified, especially when you're talking about treating young children that are going to have many more modified cells and maybe have them for many decades. So we worry about double-stranded breaks. So gene editing is one way that gets you around double-stranded breaks, and one this is from a paper from Mitch Weiss's lab at St. Jude's, which is I think really cool because it looks at a way to exploit gene editing, making a single base pair changes in the human genome to fix the sickle cell disease genotype. The way this works is that we're going upstream of the gamma globin promoter. This is the same gene that gets alternatively regulated by BCL11A, but it introduces single mutations that. So the way to think about this is that when you have a promoter or an enhancer or upstream regulatory elements, they'll contain motifs that can bind repressive transcription factors or activating transcription factors. And these are some clever mutations that are actually naturally occurring in humans that oftentimes change a repressive to an activating transcription factor. So here we have. Here we have a KLF1 motif, which is activating, and I think here we're over here we have a BCL11A motif, which is repressive, and we're changing for a GATA1 motif, which is activating because GATA1 likes to activate globin genes. So very, very elegant, and importantly, we know that these mutations are called HPFH for hereditary persistence of fetal hemoglobin. And we know that if you have two copies of sickle globin, right, so sickle cell disease, plus one HPFH mutation, you don't have sickle cell disease. So if you can install one of these changes, you could potentially cure sickle cell disease. And this is actually, preclinical, very nice preclinical data in this paper from Mitch Weiss's lab. So they installed one of these HPFH mutations, and you can see that the fetal globin percentage compared to untreated cells went up by a lot. And this is taking CD34 positive hematopoietic stem cells from patients with sickle cell disease. Very, very cool. So this is something I think is really interesting. I'm not saying this is exactly what we're going to do, but this is certainly very interesting, and we're exploring this kind of general approach right now in our lab. The other thing that we're exploring is to make these genetic changes not with electroporation or with viral approaches, because in the case of lentiviruses, they're very expensive, take forever to get a GMP batch of lentivirus produced. And electroporation really beats up on these cells, and these come from patients whose cells are very difficult to collect, sickle cell disease, patients, you cannot give them GCSF. It's contraindicated because there have been cases of splenic rupture and death. So you can only mobilize them with plerixafor, which is pretty awful. You get very few stem cells. And then, we send out these stem cells to our colleagues, and then we get very few stem cells back after they've undergone electroporation. So LNPs are a way of getting around that. So these they have actually been widely used in COVID-19 vaccines, most famously, where they've delivered mRNA to over 1 billion people, and really everybody who's gotten a COVID-19 vaccine has gotten temporary gene therapy because they got mRNA that instructed cells to produce a spike protein of the virus. So LNPs ought to be they're widely tested. They should be pretty efficient, and we should be able to safely deliver genetic materials without using viral vectors or electroporation in an ex vivo approach. So here's sort of the idea of how we want to get towards cost-effective semi-automated gene editing for sickle cell disease, and this is my last slide. So this is in collaboration with Eugene Kandros and Mohammed Gabriel Alame, Steve Grubb, and Yongping Wang. The idea would be that we manufacture clinical-grade LNPs in-house, and then we use those LNPs to manufacture gene-edited hematopoietic stem cells for sickle cell disease also in-house. And then we're starting up a research collaboration with Miltenyi that I'm super excited about, where they'll build a version of the HSC protocol that can take LNPs instead of lentiviruses. So we can make these on the Prodigy, and then we could take this protocol anywhere that has a Prodigy, which is super, super cool, I think. And we're very interested in partnering with other industry partners to bring the cost for effective sickle cell disease gene therapy products to patients. So if anybody wants to talk to me afterwards, I'd be very happy to chat. So this brings me to my acknowledgements, folks from CHOP, from INCA, and from UFRJ, which is the Federal University of Rio de Janeiro. And I'm happy to take some questions.